Welcome to this session of the World Stem Cell Summit entitled The Amazing Reverse Migration of Products from 361 to 351. I want to thank the World Stem Cell Summit led by Bernie Siegel and the Regenerative Medicine Foundation for this opportunity to present today. Today we'll be talking about the implications of FDA's announced end of enforcement discretion on May 31st of this year for products that previously were marketed without FDA approval as human cell tissue and cellular and tissue-based products regulated solely under the authority of Section 361 of the Public Health Service Act that FDA now believes do not qualify and now require prior approval from FDA. We've aimed this session toward those who make, sell, administer, and use products that will require some form of FDA approval. And for this session, we focused on products that will be biologics requiring licensure under the Public Health Service Act. There are some products that may qualify as medical devices. If you have those products, or you may, feel free to come talk to us separately. And we'll also not be focusing on whether or not your products qualify. For this session, we'll be focusing on those that will actually require some FDA approval. If you are not sure, again, feel free to come consult us. But today we'll be talking about the challenges of making that conversion from a product previously marketed without approval to a biologic requiring FDA licensure. So I'm very pleased to have with us one of our speakers, Dr. Rebecca Brown, who's the Vice President of Global Regulatory Affairs for MiMedx Group, Inc., which is a biopharmaceutical company developing, manufacturing, and marketing regenerative biologics utilizing human placental allografts for multiple healthcare sectors. Her responsibilities include executing regulatory strategy and advancing regulatory approvals for MiMedx products worldwide. Dr. Brown has over 20 years of experience leading teams to produce innovative and impactful products. She has an impressive track record for integrating customer needs with company goals. Her unique combination of engineering rigor and business acumen have not only positioned her company and its products for success, but are shaping the regenerative medicine field for purpose-driven focus on how to best help patients. Dr. Brown holds a PhD and an MS in mechanical engineering from Georgia Tech, where she was an NSF graduate research fellow and has a bachelor's degree in mechanical engineering from MIT. She's the inventor or co-inventor on multiple US and international issued patents, serves on the GT School of Mechanical Engineering External Advisory Board, volunteers with MIT, and is active in multiple industry organizations. I'm also very happy to have my colleague, Jane Clement, with us today. She is a senior associate in the Washington, DC office of my firm, Hogan Lovells, part of the FDA pharmaceutical group. By background, Jane is a clinical PharmD and a JD with a particular interest and specialization in regenerative medicine, which includes gene therapies and complex biologics, assisting both smaller and larger companies trying to navigate FDA's complex regulatory framework. I will be moderating and discussing some of the things today. I'm a partner at Hogan Lovells in the same group, the FDA Pharmaceutical Group, and I chair and founded our firm's global cell tissue and gene therapy working group, which has over 140 members around the world. So with that, we'll turn to our presentation. Next slide, please. So I just want to emphasize that today we will be individually expressing our own views and not those of formally those of Hogan Lovells or of MiMedx Group, Inc. Next slide, please. All right, so I wanted to start with a basic overview of the regulations involved here, and talk about just a quick framing of how we got here, which Jane will help me discuss. So first of all, we have what are these products? Human cells, tissues, and cellular and tissue-based products are defined as human cells or tissues is intended for implantation, transplantation, infusion, or transfer into a human recipient. Historically, they were regulated in a piecemeal fashion in many different ways, under the practice of medicine, as human tissues for transplantation, or other frameworks. And finally, in 2001, FDA determined that it's necessary to consolidate and finalize a risk-based regulatory framework for tiered risk-based approach to the entire class. There are 4 criteria. In doing so, they decided that there will be some requirement for all of these products to prevent the transmission of communicable disease, and that's part of the authority that FDA had under Section 361 of the Public Health Service Act. But in doing so, they determined that some products do not, based on a risk, do not really require pre-market approval or review. And if they meet certain criteria, which we won't go through in detail today, because that's a topic for a different discussion, then they could be on the market without prior FDA approval. They did still require inspections, and they must register with FDA and list the products, but there were not any pre-approval requirements. So next slide, please. So how do we get there? I'm going to turn it over to my colleague, Jane. And if you could talk us through briefly, how did we land where we are today?
Well, thank you so much, Mike, and thank you for that great introduction. I guess before we get into this about this great reverse migration, we have to understand, well, how do we get here that there needs to be this great reverse migration back to 351? So essentially, what ended up happening since 2001 is that the field blew up. It has really just progressed rapidly through the practice of medicine. These products are used widely. And essentially, the way we think things kind of evolved is that this process, in terms of classifying yourself as a 361 product, is self-designated. Really, there is no FDA okay or checkpoint to kind of give you that designation, but companies did this. It's a self-designated pathway or process. So because of this, and because over the years, really, there was very minimal and low enforcement from FDA, there was really a lot of confusion in the field and industry about, well, how do you interpret those 4 criteria to qualify? So generally, the field proliferated. It's widely used, and here we kind of are today. But then 2017 has come up. So in November, FDA announced a brand new regenerative medicine framework, announcing its intent to go ahead and enforce the pre-market requirement. FDA not only clarified the criteria, they issued a series of guidance documents, which included one of them that talked about the 4 criteria and FDA's kind of expectations and recommendations around those. But they said that they will be giving everybody about 3 years of enforcement discretion to assess the criteria to determine if they qualify for 361 status or whether they need to file an IND and seek pre-market application and approval. That enforcement discretion was extended due to the public health crisis with COVID-19 by 6 months. However, that deadline in terms of enforcement discretion, which was risk-based and applied to more lower risk products generally, expired on May 31st, 2020. A lot of us were hoping to get kind of more guidance or clarity around whether or not there might be more extensions around this enforcement discretion policy or compliance window. But as of earlier this month and towards the end of May, FDA did announce publicly through 2 articles, one of which was a FDA Voice article published by Dr. Peter Marks, confirming that no further extension will be provided. FDA not only confirmed that the extension will not be extended, however, they said that if manufacturers continue to market these unapproved HTTPs, they do so at their own risk and will be subject to enforcement action, such as warning letters, untitled letters, and possible court dispute resulting in injunctions. The article, what was kind of interesting for all of us, is that FDA is now publicly kind of announcing some of the categories, even though this list is not exhaustive, as to who should be on alert. This might include stem cells, stromal vascular fracture, umbilical cord blood and or stem cells, amniotic fluid, Wharton's jelly, orthobiologics, and exosome. So essentially now here we are kind of knowing that there's going to be these standards that are going to be applied on all these products that traditionally were marketed without approval. So, I think I'm going to, Mike, it would be great if I can turn to you to kind of walk through what a company is going to expect with now these new standards that will be applied for them.
Thank you, Jane. If we could go to the next slide. So these products that no longer qualify are essentially unlawfully on the market unless they have some form of FDA approval or permission. So the basic way that one would get a biologic licensed is to go through the full approval process with FDA. The history is that biologics, unlike drugs, were originally regulated by the National Institutes of Health. And so the licensure process derives from a different statute, the Public Health Service Act. And there was a particular provision, Section 351 of the Public Health Service Act, that puts forth the standards for licensure in order to get approval to market a biologic. Interesting enough, the provisions are very succinct. They don't have a lot of detail. And as you can see on this slide, the basic provision is that to get approval, the Secretary of Health and Human Services, it's been delegated now to FDA that took over jurisdiction from NIH, shall approve a biologic license application, a BLA, on the basis of a demonstration that the biological product that is the subject of the application is safe, pure, and potent, and the facility in which the biological product is manufactured, processed, packed, or held meets standards designed to assure that the biological product continues to be safe, pure, and potent. Seems to be pretty simple. And then there's delegation of authority to FDA to promulgate regulations in order to, and it doesn't give a lot of guidelines or guidance. It's basically the secretary shall establish by regulation and requirements, the requirements for approval, suspension, or revocation of biologics licenses. So in 1997, there was the FDA Modernization Act came along, and there was concern that there were differences between licensed biologics and drugs, pharmaceuticals approved by FDA under the Food, Drug, and Cosmetic Act. And so Congress passed a provision that instructed FDA to take measures to minimize the differences and to bring licensure of biologics to be as close as possible to the same for drugs. And that's really what's really at play here, is that FDA historically, since then, and actually even before, has treated all products that require licensure like drugs. And in fact, except for that small category of products that are medical devices, all the provisions of the Food, Drug, and Cosmetic Act for drugs also apply to biologics. And so except where there's a specific difference, like the licensure process, where it's different language and it's named licensure, a different provision, otherwise all the requirements of drugs apply to biologics also. So if we could have the next slide. So I won't go into great detail, but there is a regulation that sets forth kind of at the high level what's required. And this slide here summarizes it, but it sets it forth at a very high level. And you'll see, for example, the fourth bullet down says a full description of manufacturing methods. So that is very misleading, and it sounds very simple and perhaps easy to fulfill. But packed into that language is the world of requirements, both set forth in the regulations, largely in Part 600 of Title 21 of the Code of Federal Regulations, and a huge history of guidance documents and approvals, regulatory approvals that have set forth what the standards are for the kind of data required to get licensure. So if we could go to the next slide. And because biologics are drugs, FDA has imposed a lot of the formal regulatory requirements for drugs on biologics, including the content and format for an actual BLA, biologics license application. Now, these are these applications that a new drug application is an enormous set of documents that can cover like an entire wall if printed out. And in the there's in the Code of Federal Regulations, there's almost 11 full pages just describing what that application has to entail. And here's a summary list of those things. Not all of them apply, but many of them FDA has applied to biologics, such as non-clinical pharmacology and toxicology data, even before one begins to test it in humans. So we could go to the next slide. Here is the form that goes on top of that enormous application, Form FDA 356H. And in fact, it's the same exact form that governs drugs and biologics. And you can see there are references in there to both Part 314 of the Title 21 of the Code of Federal Regulations and Part 600. 600 is the biologics, 314 is drugs, but also applies to biologics. So this gives you an overview of a lot of the similarities. Not all of them apply to biologics, but most do. Next slide. So here are a few similarities. We won't go into detail, but a few additional similarities. An important one is the investigation of new drug regulations. So in order to test a biologic or a drug in humans, you have to get essentially permission from FDA under what's called an IND, investigation of new drug application. And there's a whole set of regulations that governed drugs before FDA got jurisdiction over biologics. Those were all basically applied to biologics. So before you apply for approval to FDA, you have to go through a whole testing regime in testing your biologic in humans under this scheme, under this IND scheme for that also applies to biologics. And there are a bunch of others here. We won't go into detail, but a lot of similarities, including some preferred treatment for speeding up the process. And we've listed those that apply to both here: fast track, accelerated approval, priority review, and breakthrough therapy. We can talk to you separately offline about the details of those. Next slide, please. A few other similarities that make this process actually quite long and challenging and expensive include some of them listed here. I'll just point to two. One is when you finally finish your testing and you're ready to apply, there's actually an application fee, which is almost $3 million. If you for the first time when you're submitting clinical data. There's also annual fees that are not insubstantial either, that go on every year. They get adjusted, but they're also usually in this general range of $336,000. So it's an ongoing, very substantial endeavor and investment. And another thing that makes it very rigorous and expensive is that biologics as drugs must satisfy the current good manufacturing practice regulations to make sure that the manufacturing process is can produce the kind of quality that is necessary. Next slide, please. So what's unique about biologics? There are some things. First of all, the verbal standard is actually slightly different. For drugs, it's safe and effective. For biologics, you have to show that the products are safe, pure, and potent. Now, the standard turns out to be somewhat similar, but there are some differences. And there's regulations that define it. For example, pure purity for biologics, it tends to be a lot more complicated because they're typically derived from the body. And so they can't be sterilized to the same degree that small molecule drugs can usually. So the definition is relative freedom from extraneous matter in a finished product, whether or not harmful or deleterious. There are a few other things that are unique. Potency is determined to include effectiveness if it's the specific ability or capacity to affect a given result, which can in some cases be a little bit different than for drugs. There are a few other differences we won't go into detail. There's a special designation for regenerative medicine advanced therapies, or RMAT. There are specialized inspection teams. There's a philosophy that the process is very important, and that because sometimes the end product can't be as discreetly characterized as small molecule drugs, that's very important to control the process, because the end product might have a certain amount of acceptable variability that you can only ensure will produce the kind of process you need through good control over the process. And there's also, we won't go into it, but there's a biosimilar pathway for biologics, which is a bit different than the generic drug process for drugs. So next slide, please. So all that translates into a pretty significant challenge and requires a lot of investment and usually quite a bit of time. So I'll turn to back to my colleague Jane to summarize for us what some of the key expectations and what generally it takes at the high level to get approval.
Sure. Thank you so much, Mike, for that kind of overview of just kind of the logistics of what are the distinctions and what are people now, what should sponsors expect? Well, another really big part of this, and I think a lot of people are thinking about this, is how long it actually takes to get approval or to develop a BLA. And the process, can take a substantial amount of time. The Government Accountability Office recently reported that sponsors on average take 10+ years just to develop their product from the clinic to approval. And it can cost up to sometimes $2.6 billion just to kind of bring a biologic or a new drug entity to the market. And generally this process involves the clinical testing. I mean, there is a lot of non-clinical, pre-clinical work that goes into it, but I'm going to cover just really high level what people should expect in terms of the 3 phases, or what you're typically seeing when you develop your product. So first, you would definitely have to do phase 1 trials. And this is generally to determine how the drug or the biologic interacts with the human and to see about side effects with regard to the dose escalation or to pick the right dose. This is typically done in small patient groups, maybe 20 to 100 patients or so, and usually sometimes healthy volunteers. And this could be several months, but it could be longer, but that's just typically the timeline. Next, the product would enter phase 2 trials. These are larger trials and much more costly usually, where hundreds of patients are studied. And this is where you start to really ascertain the product's efficacy to see, could it work for that particular indication? And what are the short-term side effects or risks that might happen? This data typically kind of refines your research question in order to, before you make the large investment into phase 3 trials and to potentially gather the real substantial evidence that you need, it'll help you kind of focus your data into hypothesis setting and to develop your phase 3 trial. So oftentimes an end of phase 2 meeting is done with the FDA, where you're going to essentially align on what is needed for your phase 3 trial in order to go ahead and proceed with that development. So generally, a lot of sponsors really take advantage of this meeting to really form their development program moving forward. Generally, that process, just in terms of going from phase 1 to kind of end of phase 2, could take years, sometimes 3 to 5 years or so before you actually even get into the clinic to do your pivotal testing. And then for phase 3, generally FDA requires up to two phase 3 trials to prove substantial evidence of safety and effectiveness. And here you really are testing this in 3 to 3,000 or thousands of patients, because that number varies depending on your statistical plan and what's needed in order to actually get the evidence that you need to seek approval. But these trials are really, really substantial. So a lot of times you really want to get feedback from FDA to make sure you're aligned on what to expect before you conduct your phase 3. And phase 3 testing, just for a single trial, could take up to 1 to 4 years just to complete one, and sometimes more than one is required. So I think it's just really important that just to have this as our level setting, to hear from Dr. Brown and kind of industry perspectives on what could be some of the challenges with going through this clinical development process now that you have to transition from 361 to 351 while you were previously approved. So with the next slide, I'm going to turn to Dr. Brown to kind of walk us through some of those. Next slide, please.
Thank you, Jane, and thank you, Mike, for the introduction. I guess first off, I want to say, I'm going to talk about a lot of challenges and a lot of difficulties that need to be overcome. But just out front, I want to say that we do support what FDA has done here. We do realize that there were products that may not have been so safe on the market. And so we definitely support the FDA's role in this and their intention to kind of clean up this area of the regulatory space. So these are just challenges we'll discuss. They're not complaints, they're just opportunities to work through. So I just wanted to get that out there out front. So of course, from a company's perspective, change is hard. And there are many challenges that we are facing as we move a product from a 361 category to a 351. So even though, of course, we are very focused on our manufacturing process, our testing, our tissue sources, all of that, we really have to remember that the true focus should be on the patients that are using and being treated with these products. So of course, in some cases, these products have been on the market for over a decade. And the physicians and the clinicians using these products have experienced great safety and efficacy with them, of course, in some cases. And it's very difficult for them to transition as well, because one day they were able to purchase the product, and then June 1st, after enforcement discretion ended, they couldn't purchase the product at all. So it's significantly impacted their practices, and we really should be doing all that we can in collaboration with FDA to get these safe and effective products back in their hands as quickly as possible. So of course, a traditional way to do this is to work through the BLA process. But as you've heard, and as you know, this can take significant amounts of time, years and years. But while we do this, we should also explore other avenues in parallel. How can we work with the FDA to determine if there are other options available, other avenues to explore? Is it expanded access? Is there something else available or something else that can be developed with FDA to get these products get the products that are safe and effective back to patients that truly need them and have used them for so long in the past? So there's no easy answer to this, no quick solution, but we really have to try. We really owe it to those patients and clinicians. So while we're very supportive of FDA's position, there are challenges for us. So one challenge is, obviously, there are products out there that FDA feels are not 361, but classifying those products into their new classification is not always transparent. If you look at the industry, we see structural tissue allografts that are on the market as devices, and then there are others that are going through the BLA process. So it's not always clear to others in the industry how that happened or why that happened, or was it a claim, was it a mechanism of action, was it the review committee they received, was it because they pushed a certain way versus pushing a different way in conversations with the FDA. So it would be very helpful to the industry if there's additional transparency on the classification process. Of course, there's the RFD process, there's the TRG opinion that companies can receive, and there's guidance as well. But even with all that information, it would be very helpful to have additional transparency and guidance from the FDA in this area. Also, and I think this is really a bigger issue, most of these companies that are transitioning products from 361 to 351 are doing this for the first time. I don't know if anyone's actually transitioned a product completely yet. So we're all in it together. And there's really no precedent to follow for structural tissue allografts. Of course, there are other products that are licensed BLA products, but none of them are human tissue allografts that used to be on the market as 361. So there's really no one to follow, and we're kind of, feeling this out as an industry as we go. And of course, interaction with FDA, as much as you possibly can, is critical to the success and the efficiency of moving through the process. And of course, that's even more of a challenge now with COVID-19. And FDA, of course, is extremely busy doing extremely important work at CBER along those lines. So it's challenging to get the time with the groups at FDA that you need to have those conversations and move your products and projects forward. It would be wonderful if FDA could issue some product-specific guidance for tissues in this category, specification development, any type of developmental standards or expectations for what they're looking for these types of products. Of course, it's very difficult for anyone to do, or even for us to propose topics, because we haven't been successful. We haven't made it through this process. And so I'm guessing that, probably after a few products have made it through, it'll be a much easier ask, much easier project for FDA to take on to begin drafting guidance in these areas. Next slide, please. So I mentioned, of course, there are challenges, and there aren't just a few, there are plenty of challenges. So one example is the tissue, the raw material that we start with, is not something that was manufactured or synthesized in a lab. It comes from nature. It was created in a human body. And so we know, in our case, birth tissue, we only accept donations that have produced healthy, live babies. We don't accept anything else. And so we know that tissue for its job, its function, its original function, was successful. It did what it needed to do. So I guess I'm jumping a little bit, but how do you then qualify what's a failed product if you validated your manufacturing process and you need to set a specification? So the specification setting is very difficult when you know that donor tissue functioned appropriately, yet you've got donor variability simply because it's a natural occurring material that comes to you, in individual batches from a single donation, single recovery. It comes in different shapes, sizes. Of course, we accept tissues from a single birth and a triplicate birth. So we've got, an amazing variability in raw material that is coming to us. As Mike described, there's purity, potency, and identity is how we look at it for a kind of release testing or investigation we must do before we can release product. And those are new terms to us when we've grown up in a good tissue practices space. Good tissue practices, of course, were complying with a lot of the good manufacturing processes, but the real focus is on potential transmission of communicable diseases. So the purity, the potency, and the identity of the product, seem obvious. Of course, it's all placenta. Of course, or human birth tissue. Of course, it's potent. It produced a live, healthy child. And of course, it's birth tissue. That's the only thing we collect. But that's, of course, not adequate whatsoever for a BLA submission. And so there is significant work that has to be done to identify the appropriate test, develop tests. A lot of these things don't exist. And so that's where the communication with the FDA is key. What are their expectations? What are their ideas and suggestions? Of course, you have to present your ideas to them as well to give them something to critique or to give you information on or thoughts on. And again, I mentioned the specification setting. That's very difficult when you may truly believe that as long as your manufacturing process is followed and it's a validated process, that all the products should be effective and safe product. So on that bell curve of criteria you're measuring, what's a failure and why is it a failure? Should we just, discard 5% that's on the low end of the bell curve? What does that mean clinically? Is that critical? Of course, you want to be able to catch manufacturing errors, but you should also have a robust system that catches it before it gets to final release testing. So all of these are significant challenges that manufacturers have to work through as they transition from 361 to 351. Next slide, please. And of course, the challenges don't end there. One thing that is, I think, a slightly unique challenge is the batch size. So for instance, we're looking at birth tissue, and depending on the product we're making or the size of the product we're making, of course, there's a limitation of how much you can make from a single donation. So, with a good tissue practice policy or a 361 product, you definitely do have retains, you have testing requirements, but it's insignificant compared to the BLA release requirement. So for instance, if we followed the regulation exactly as written and exactly how we interpret it, if we say have a batch size of 3 processing a donor, 15 of those units would be allocated for lot release testing. That's identity, sterility, particle size, residual moisture, endotoxin, elemental impurities, residual processing agents, potency, along those lines. Then 12 of those units would be reserved, archived for testing in case anything came back on the actual samples you released for commercialization. So in that example, there are about 3 units that are released from a single donor. And so obviously that doesn't make a lot of sense in terms of commercializing a product from a small batch. So that gives a great opportunity to work with the FDA to understand what can we do in that case? Are there byproducts of the process that we can use for some of those tests? Are there smaller sample sizes that we can use with perhaps a advanced modification of the testing process so that you can use a smaller sample size? There are lots of ways to look at that challenge to try to reduce the number of units that actually have to go to test and the number of units that are retained while still meeting the requirements of the regulation and accomplishing all those tests.
Rebecca, I'd like to just interject for a moment and just point out that it'd be a lot of people say, why can't we just get FDA to tell us the answer? What is enough? Or can't we just do it this way? And FDA, a lot of people don't really understand that FDA does not always know the answer. FDA is a data-driven agency. And so it's up to sponsors, applicants, to provide the data to FDA that it can rely on. FDA, it has some labs and it does some of its own research, a little bit for quality control, but primarily it relies on what it receives from applicants. And so there is an opportunity here. There's a challenge because in some ways we're operating in the dark and we don't really know what FDA will find acceptable. But on the other hand, it's an opportunity. It's an opportunity to present data to FDA that will demonstrate that we could get away perhaps with fewer retained samples, for example, but you have to prove it to FDA and to convince them. So there's a certain, there's an attitude of advocacy that's necessary here also. Certainly good scientists, good science, and that foundation is important, but it's also important to realize your audience and that FDA views itself in a way more as a judge than as an active innovator. Now, there are some exceptions where they're pulled into the process, and there are ways to engage FDA to have more of a back and forth process. And that's part of some of the programs that we mentioned, the RMAT, for example, but the historical position is that they're more judging your data. So it's important to think in terms of that advocacy when you're designing your programs and figuring out how to present it. So now I'll turn it back with that additional flavor. I'll turn it back to you, Rebecca, to talk about some more of the challenges that companies will have to advocate to FDA about.
Thanks, Mike. And I couldn't agree more. You know, for instance, on the sterility testing and retains requirement, if you look at that from a different perspective, maybe outside of CBER, it was shocking that they do require lot release testing for sterility. And it's because our product is terminally sterilized. And so I think the regulations were focused on products that you had aseptic processing with no terminal sterilization. So of course you would have to require a lot release sterilization test. But if you actually dig into the science and you look at all the information available, you know, we have a validated terminal sterilization process of 1 in a million, you know, SAL of 10 to the minus 6. So, you know, a chance of 1 in a million of our units being non-sterile. Well, you can't test that out of a batch of 30 units. There's no way to get to that assurance level, no matter how many. Well, you could test all of them, but then of course you would have no product left. So that's something that's written into the regulation in a certain way. They do allow for a exemption or an exception from that, but it is written that it needs to come from the director of CBER with your BLA application. You know, that's something you ask for in a BLA application. So of course, that's something we're highly interested in doing. So that's just an example of where we're challenged because, you know, we're trying to fit the regulations that we have to follow to a product that really wasn't conceived when the regulations were formed. So it's, again, an opportunity to work closely with the FDA. And of course, kind of that, this is really the potency question, the mechanism of action challenges. So if our products are extremely complex, they seem so simple, yet they have hundreds of regulatory proteins in them. They're not a single drug substance, you know, single growth factor product. So it's a challenge to sort through all of that information. What can we measure? How can we measure it? How reliable is it? How much variability is there? Is what we can find and measure that's relatively consistent, is that an important growth factor that relates to a mechanism of action that may have some relevance to that clinical application we're looking at? That's a very long, complex process. And then, of course, what happens if you add an indication and add an IND with a new indication? Is all the work that you did finding that MOA, does it apply to your new indication? Can you justify that? Can you argue that? You know, what does FDA think about that new indication and your potency testing? Is it applicable, or do you have to start over? It's a very difficult thing to go through. And again, like we've said, FDA doesn't necessarily have all the answers. They can't say yes or no, or, oh no, you skipped from A to C, you need to do B. They're living it just as we are. So it's definitely a challenge. Never a dull moment. So on top of all of that, like we've talked about before, we have had use of these products in the market as 361s. And so in some cases, we have 10 years of experience of patients and clinicians using these products. And so there is value, information out there that has some value. So trying to understand how that fits into the application that we'll eventually file. How can we use that information that is in a way that FDA believes is appropriate for the application and for a BLA filing? You know, in some cases, hundreds of thousands of patients have been treated with these products. So how can we leverage that information? It's a very, very interesting question, and I think one that FDA is also interested in exploring. And so, Jane, I think the next slide is related to that.
Just before we go on, you can go to the next slide. I just want to interject, and thank you so much, Dr. Brown, that some people have said, why is FDA doing this? And these products have been in the market for so long, and there's been no real safety events or concerns. And why is FDA doing this and insisting on this higher level? And I would say the FDA's response is likely several-fold. One is that FDA said, well, we actually don't know that the amount of data that we get voluntarily reported from use out in the marketplace is actually very poor. And sometimes there have been cases where drugs have been in the market for a decade, and only after then was it discovered that they actually have very serious adverse events. And so FDAs would say, until you study a drug under controlled circumstances, we really don't know. And so the products that have been out there for all this time, unless we really study them carefully, we don't know for sure. And so, but why do we need to know that much? And I think FDA would say, this field has great potential. And we really believe in this field. And there's so many possibilities for both curing diseases and for improving people's lives that we think that if we have better understanding under controlled circumstances, that it will propel the field forward. It will give both doctors and patients greater confidence, but more importantly, greater understanding of how and when these products can best be used. And some of the things that we don't know about it will come to light. And, you know, for the 361 framework, typically you can't use it to cure a disease. That in most cases, that isn't what's known as a homologous use or something that would qualify. And so to prove that it actually can be used to cure a disease, to treat a condition, you actually need to have real data to show that. And so those are part of the reasons that's driving this. And there, you can debate the extent, and that's largely where we're going to be focusing going in these years coming forward, is how much certainty, how much data, and how much do we need to know in order to feel comfortable that they should be then considered fully marketable again, and how can we get the most out of this process. So I just wanted to fill in a little bit of that background, and it frames a little bit of what Jane will now explain about the reliance on real world evidence and real world data. So Jane?
Yeah, thank you so much, Mike. And just to kind of even shadow what you said, but just to add, also it's because the field is very varied, right? The amount of products that are available could be simple, where it's just a shaping or processing that's a simple process, and then more complex HCTPs and regenerative medicine products where, you know, they might be relying on the body's effects of other cells or tissues. So I think what you said and what Rebecca said is just right. It's just FDA's, you know, struggling to find that balance. And hopefully this does segue nicely into hopefully some of the real world data and evidence could help the agency with making that decision and kind of finding the right balance. So I just wanted to briefly, you know, since Rebecca talked about, you know, the fact that all of these products have been on the market, or not all, but some have been historically used for years, have been kind of admitted to patients, used widely by doctors. Well, could you leverage any of that for your applications moving forward? And FDA's policy on real world data and real world evidence has been evolving for quite some time now. One thing I just want to level set, what is real world data and what is real world evidence? Those two terms are actually treated very differently by the agency. Real world data is just what it sounds. It's data. It's the historical use. It's data related to the health status or outcomes collected from, you know, real world sources. Oftentimes, what comes to my mind is a lot of times like electronic health records, patient registries, things of that nature. But real world evidence, that's a little bit different. That is actually clinical evidence regarding that same use, where the inputs is real world data. However, it's actually conducted in a study design. Again, a really good example is observational studies, whether prospective or retrospective, where you're actually going to use these data inputs that you get from health records, patient registries, various different sources to actually conduct a study or actually run an analysis with the report, with statistical analysis and plans. And really, a lot of times it can be really robust and could include data points from thousands of patients. in order to kind of come up with a real world evidence. So I think that that's just kind of to level set, because the term is used widely. And I think sometimes people say, oh, I have real world evidence that my product worked. Well, that might be that you have data or historical uses of that product, but real world evidence does require an actual study and an actual analysis, and would be kind of typically scrutinized by the agency. I also think it's important to know kind of where we started with this use of real world data and evidence, and where are we today. Historically, this was really just used for purposes of safety monitoring and adverse events. That was really when your product would get approved, all of the uses or side effects would be reported to FDA, and that could impact FDA's decisions on pulling a product or changing a product or changing a label. So real world evidence and real world data historically was really all about the safety. And FDA now has one of their largest programs with the Sentinel system, which is a very large monitoring system that's set up. However, in 2016, Congress passed the 21st Century Cures Act. I won't get into the details of everything that was included in that act. It was really a large, big piece of legislation that changed a lot of things for this just field generally. But one of the provisions in the Cures Act was that it placed a focus on the use of real world data and real world evidence. And specifically, Congress is urging FDA to support regulatory decision-making such that they said FDA has to create a program that's going to evaluate whether or not the agency can use this data to support approvals, specifically to for products that are already approved. Could the real world use and evidence of that product off, for another use or off label be used to expand the indication? Now, FDA recently, about in 2018, announced that that program, or its development program to try to assess if they can go ahead and use this type of data, is going to also apply for biological products, which is really promising, at least for this space. But generally, I would say if where we are today, we are a bit behind with are we using real world data and evidence the most effective way that we can? Because in terms of proving efficacy, we are seeing it being used in the safety space, but showing efficacy, we do not see this as often so far. There are some examples where FDA is starting to kind of use real world evidence and data for purposes of efficacy related decisions. For example, the agency uses CMS data in order to do comparative effectiveness assessments of preventive vaccines for influenza or the herpes vaccine. But generally, we've really only seen this done in the orphan drug or rare disease space and oncology, where an example would be you have a single arm interventional study where, for purposes of logistics in terms of orphan drugs or ethics, it would be unethical to have two arms or give a placebo, drug. And in those instances, with these single interventional studies or single arm studies, sometimes a sponsor is able to do a real world evidence study where they're going to assess data points from health records, electronic sources, patient registries, to create the historical control against which to compare their interventional study. And in that regard, we've seen under accelerated approval, a few examples of where there were approvals with this type of strategy. We've seen it in an oncology space in terms of where thousands and thousands of records were possibly used to expand some labeling information for new patients and different uses. So in that regard, we are starting to see it, which is promising. However, it is pretty right now uncertain, and it's evolving. So we're all kind of waiting to hear how we can best leverage this type of information, especially for this space, because these products, like you said, Dr. Brown, some of them have been on the market for years. Some of them are widely used, and if they're shown to be safe, can we kind of create study designs to create real world evidence data that could then be leveraged to speed up some of these approvals or to expand indications. So with that, if you can go to the next slide, I think it's kind of important to talk about, well, what can FDA do to help the field? Because, we've kind of told everybody about the challenges, and I think it's equally important that we just kind of spend some time to talk about what are ways that we can move this forward and what can FDA do to help. So Mike, I'm going to turn to you if you want to kick us off.
Thank you, Tim. Yeah, so one of the things that has been historically very helpful is product-specific guidance. And in the gene therapy area, for example, FDA has done that. In many cases, it's come after some approval. So they've gotten some, they've had some experience with particular products, but it's definitely something where industry and the FDA can and should work together to try to decide what they can rely on that already exists, and then what else will be needed for FDA to come out with some more prescriptive recommendations, something to guide industry. So that's one area that we think would be helpful for industry and including patients and physicians too. We want to get the input of all interested party to help make judgments to in that where we're talking about that place of judgment about how much do we need to know. Getting the input from doctors and patients is very important also. And there are some particulars that Dr. Brown talked about being challenging. And I'll just touch on one that Jane alluded to earlier, which is potency. It's a statutory requirement that products be shown to be potent, to be able to affect a certain outcome regularly and consistently. And for these products, that's a very big challenge. As Dr. Brown mentions, there's very complicated mechanisms of action, and FDA expects the potency assays to reflect how the products work. So it's often not enough simply just to take some random characteristic of the product and say, listen, we've retained it. We've gone through the manufacturing process. It's still there. FDA wants to see some relationship between the actual mechanism of action and the working, the effectiveness of the product, and the test that you do. So that, for example, if something during manufacturing happens that you don't know about that damages the product, and it no longer actually can still affect that result, the test will show it. And that is very challenging with these products. There was a citizen petition that was submitted and was decided in May of 2020, where a company requested that its real-time glutathione monitoring methods be used as a standard potency assay for all cells, because it's a common process of many of cellular products. And FDA came back and denied that on the grounds that it's not enough generally to decide that this is a process that's true for all cells. It's important for every particular application that it be relevant. And so they didn't reject it, but they said, we need to see evidence and you need to convince us on more on a product by product basis. Now there's challenges to that because that could become very expensive. And one of the areas is whether FDA can be convinced that there are broader categories where potency assays, for example, may apply. And that's been done in the past, including for cellular products, but having FDA weigh in on some early submissions of data to get FDA's view can be very helpful to companies to help prevent them going down long paths of developing assays. And this can take a long time sometimes that turn out not to be satisfactory to FDA. We want to minimize those circumstances. So now I'll turn back. I know you talked a lot about real world evidence and data, Jane, but were there any other things that you'd like to add about what FDA can do here?
Sure. Only that I think, the framework FDA's been thinking about with these products was kind of before this, the big transition, for some of these products that have been in the market. So I think specific guidance from FDA here about specifically how HCTPs could leverage some of this would be really helpful, specifically on the study design. If FDA could kind of provide, maybe that kind of goes well hand in hand with the product specific guidance, but to provide concrete feedback as to how some of this could be leveraged, because like we just talked about a little bit ago, some products are complicated and complex, and some really are more simple and more just true tissue that's been kind of cleaned or sized or minimally processed, but maybe the use is different, or the use might be non-homologous, right? So if it doesn't meet the 361 criteria, but is otherwise really, really simple, could we leverage some of that historical use to support an indication expansion? So one of the ways we can envision this is a product goes through this whole process, does clinical trials, does gather substantial evidence for an indication, but some of those historical uses could then be brought into their label, or possibly get feedback from the agency about how once they're approved, they could have still more than one use, given the fact that patient access is an issue. And a lot of patients and doctors are missing these products and have used them for years. So I think a lot can be done with real world evidence, especially with regard to how many studies might be needed. And I know, Dr. Brown, you were talking about this being such a big issue with regard to some of the challenges that industry is facing. But do you think that might be one of the ways, real world evidence or data could be used with regard to the amount of data that's needed? I kind of want to seek your input on that as well.
Yeah. Can you hear feedback?
Yeah.
Is it better now?
I think that's a little better.
Yeah, that leads right into the next bullet. So basically, it's kind of understood that FDA is expecting 2 well-controlled randomized clinical studies for your BLA submission. And so given that some of these products may have extensive data from historical use, is there some real world evidence that would allow a company to provide, one well-designed prospective randomized clinical study along with that real world evidence and use that as a package?
Looks like you may have frozen. So I'll rate. Thank you so much. Oh, are you back?
Here to see if that's going to be a possibility in the future.
Okay, well, I will thank you. I'm sorry, there's a little bit of audio problems there, but I'll just wrap it up with a few final thoughts, that there's a lot of opportunity here for all stakeholders, for industry, for FDA, as well as for physicians and the patients who ultimately these products are for, to give input to help try to reach the optimal goal. We definitely, as we made clear, obtaining licensure from FDA is a long and difficult process. Many companies have started and are far along that road, but we'll need to really work hard to accelerate that process, and try to get approved products in the market as soon as possible, so the patients that have been relying on it, them and doctors all these years, will not be deprived for longer than needed, and that we'll get the information necessary to optimize these products, take off the ones that actually are shown to have safety problems, and then figure out the best ways to use these products to their maximal efficacy. And in the meantime, it's important to find ways to avoid patients being just cut off. And we alluded to the investigation of new drug application process, where during the testing process, and we won't go into detail, but there is a process called the Expanded Access Program, which is set forth in regulations that FDA has, that allows patients that don't necessarily qualify for one of these very narrowly designed clinical trials aimed at finding a very particular effect. There's many reasons a patient might not qualify, including they might have other conditions, other diseases or other conditions that disqualify them. FDA has a process for allowing patients that don't qualify for the study to get access in the meantime. And it's important that patients be made aware of that, and companies work with patients, doctors, and FDA to ensure that patients will have access, the maximum access to safely controlled uses of these products while this longer process is proceeding to determine that they can be now migrated back to the marketplace, but now with more understanding and more knowledge and better control. So with that, we'll we'll close the presentation. We could go to the last slide. And just thank again, Dr. Brown, Jane Klinina, and the World Stem Cell Summit, Bernie Siegel, Regenerative Medicine Foundation for this opportunity. We hope this is useful, and we look forward to working together to bringing this, to migrating these products to full approval and in the right time, and to bringing regenerative medicine to its full potential. Thank you so much.
Thank you.
Thank you.